Well, good afternoon, everybody. Welcome to our second podcast, the first in our Think Wheat series for 2021. My name is Dallas Carpenter. I am the Communications Manager for the Saskatchewan Wheat Development Commission. Today we will be hearing from two speakers on new wheat varieties for 2021. Our first speaker is Dr. Richard Cuthbert, research scientist and wheat breeder with Agriculture and Agri-Food Canada, who is based in Swift Current. Richard will explain the wheat breeding process from making initial crosses to the release of the final wheat cultivars. Our second speaker is Jim Downey, Research and Development Manager for SeCan. Jim will present information on newly approved wheat varieties, covering information from trials and the Saskatchewan Seed Guide, including aspects from Fusarium head blight resistance to tolerance to environmental stressors such as drought. Please note that this webinar has been approved for 1 CEU credit by the Prairie Certified Crop Advisor Board. If you are collecting this credit, please go to our website, saskwheat.ca, for options to submit your CCA number. We will have a limited amount of time at the end of the presentations for questions. Please use the Q&A function at the bottom of the screen to submit your question, which I will read. Alternatively, put up your hand during the Q&A session to ask your question audibly. I will get to as many questions as possible. That all said, let's get everything underway. Please welcome Dr. Richard Cuthbert.
As well as an irrigated environment. We don't really want that orange one where it does quite poorly in a drought and does reasonably well in the irrigated. So we're looking for reliable, high performance. As well, as I mentioned, we have evolving pathogens. Some of the famous ones are rust, and we have stripe rust, stem rust, and leaf rust pictured there. So the area of growing in Canada, Western Canada, is vast. It's a very, very large area, and there's a number of regional challenges that exist, from abiotic stresses like the number of frost-free days that we have, heat stress, drought, excess rainfall and irrigation, more of the biotic stresses or living stresses of stripe rust, leaf rust, stem rust, leaf spots, Fusarium head blight, deoxynivalenol in the grain from Fusarium head blight, and the pathogens associated, common bunt, loose smut, and then two of the pests that we really deal with at Swift Current are wheat midge and wheat stem sawfly. And kind of overarching all these is the market class acceptability that we're targeting for those crosses. So our end use quality traits, and I could have a whole slide on those, but I'll keep it simple. So I like to refer to the wheat breeding toolbox, everything from crossing through DNA-based markers, use of doubled haploidy, controlled environments, indoor growing, where we can do our crossing and marker work often, access to disease nurseries to be able to screen for these pathogens and pests, use of contra-season nurseries. We tend to use Southern Hemisphere nurseries in our winter, so New Zealand and Chile would be our primary places. And then access to adaptation trialing at the sites where we want to test yield and agronomics, as well as receive samples for grain quality testing. So you need to be able to test at multiple locations and targeted locations. It doesn't do us a lot of good if we're going to test only at Swift Current. If you wanted to release something in the Red River Valley, you need to be able to ensure that you test across many locations, and you'll see why as we go through this presentation a little bit more. But we need to be able to test at all those locations so we can achieve that minimal G by E interaction and that reliable high performance. And then the pest screening, quality testing is very expensive typically, and very extensive, as well as genomic selection is a new tool, and we'll speak about that a bit at the end, and a very new editing that's still. This is just a snapshot overview of the Swift Current Canada Western Red Spring breeding pipeline. We typically make about 24 crosses a year, and from those crosses we achieve generations of material. And the F stands for filial, and it's just a generation that we refer to as we develop inbred lines. So from the F1 to the F8 listed in the center of the screen there, and we have some numbers beside it. So we typically start with a very large funnel at the top, a lot of diversity in the F2 generation. That's where the genes from the parents are coming together. and we're trying to funnel those into the best material out of the cross. So we go from 300,000 plants in the F2 down to 8,000 lines derived from those plants. And ultimately we get to the F8 generation at the bottom, where we would go into advanced testing at about 800 lines per year. So we've gone from 300,000 to 800. And then when we get into A testing, kind of going up the side there, we can only handle about 240 lines in an A test, 25 lines in a B test. And this is annually, it's cyclical. And in a registration test that takes 3 years, we typically have about 12 lines on the go. And out of all that, per year, we average a variety, one line that came from one of those 300,000 F2s typically. So it's a longer process, and there's a lot of testing along the way, and hence why it is fairly expensive to do. So that's just really to provide some background on the numbers by generation and a breakdown of some of the generations, and we'll dive into this a little bit in more detail, but we'll keep it at a fairly high level, or try to. So this expands it a bit. The crossing can be done in various ways, but typically for wider crosses, wider diversity crosses, we would combine 3 parents in that kind of way. We cross the first 2, and then the progeny from it, we would cross again to another line. And we do that for more diverse material. For very high valued material, we may go through what we call the doubled haploid program. For high value crosses, where it would typically be 2 parents, and they would go through that DH program. The value of the DH program is you go from segregating material to inbred lines in basically a year to a year and a half. When we go the more diverse route, it typically takes about 4 years of testing and inbreeding to achieve material at the same level of inbreeding. And when I say inbreeding, I mean you take the seed from that plant and grow it again, and it will look the same. So then we go to the F2 generation, which we talked about at 300,000 plants per year, typically assessed out of these crosses. And we look for the best ones for simple traits. So you can't really assess yield, unfortunately, on single plants. Typically, you can only do what we call simply inherited traits. So leaf rust, stem rust, stripe rust, general plant type, a common bunch resistance, or some of the traits we target at that stage. Not things complex like yield, grain protein, or end use quality. To shift our odds, we will often use DNA-based markers on this material to, in the lab, be able to select plants that are more likely to be what we're looking for. So we know that the ones that don't have that gene, or the marker associated to that gene, so we discard them as early as we can. So DNA markers help us do that in the lab in a setting where it's fairly cheap and not as it's not timed with a field season. So just to put some pictures to that, this is a typical F2 space planted nursery. There's 300,000 single plants in that field, roughly. There's of course more plants than that, but we have spreader rows that are susceptible to leaf rust and stem rust to help spread disease in the nursery. But there's 300,000 roughly single plants from those crosses. And we kind of talked about it there. We look for the simple traits. So on average, one F2 plant out of that field becomes a variety per year. So some context, in 2004, this exact field, AAC Brandon came from a single plant in that field. So it's finding the needle in the haystack, really. From the DH program, the lines that come out are pure breeding. So we don't have single plants here. You can see the little rows on the screen. There's roughly 8,000 rows in this field, and many of them are doubled haploids, and the plants within the row are identical. So they're like twin sisters, identical twins. So these lines are each segregating, each row is unique, and we're looking for the same sorts of things. We can't really do yield here because we're short on seed, but we're doing simple traits like plant type, height, straw, maturity, and some of those disease resistances. And again, on average, out of the DH side of the program, on average, one DH line becomes a variety per year. And this just splits it down. I don't want to get into a lot of detail here, but it kind of gives you an idea once we get those F2 plants selected. We get F3 rows that we send to our off-season nurseries, and we go through a process of looking at these lines, and they're kind of, each one's like a family, they're sisters, and we look amongst them to find the best. And when we get to that inbred generation, that F8, we go into advanced testing, where we use more locations, more intensive disease testing, and more end-use quality parameters that are more expensive. So it's really going from the cheapest traits to the most expensive traits. So these are a few more pictures of what things look like along the way. We have yield trials. These would be run at multiple locations. You're looking at swift current here, combining. We look for more, now we can do yield on these micro yield plots, and we're able to look for the stable performing lines over years and over locations, looking at grain yield, grain protein, and other agronomic traits. And the samples from these get used for end-use quality evaluation. So the hardness of the kernel, the milling quality, the flour yield, and the gluten strength parameters. This is an example of an FHB nursery. We rely, Swift Current isn't the best place to do FHB research. We rely on southern Manitoba quite a bit. And this is the Morden, picture of the Morden nursery. And on the right side of the screen, you can see a line that's clearly susceptible and a line that's has much better FHB resistance. And that is actually AC foremost on the left and AAC penhold on the right. So penhold is a significant increase in FHB symptom resistance there. So we also use leaf rust and stem rust nurseries. There's probably, I'm guessing these are little hills. If you were to zoom in, there's probably about 20 to 25,000 hills here. We rely on the pathologists in Morden again to help us out with some of these nurseries to evaluate material concurrently that's in yield testing. So to put all this together and use a real world example, I've got Starbuck and Wheatland here, and I suspect Jim might talk about them a little bit. The crosses were made in 2008. They have pedigrees listed there. Starbuck's in the left column, Wheatland's in the right column, and they follow a very similar protocol, but where some things are a little bit different, I've highlighted them in green. So going from the cross in 2008 to essentially inbred lines in 2012, roughly 4 to 5 years, we had a bunch of lines that met a lot of, that checked a lot of boxes, but we aren't really sure of their performance until we get into that advanced testing and registration testing. And that took us the next 5 years of 2013 to 2017. So 2 years of advanced testing, 3 years of registration testing, and those lines were identified to go forward. They were supported for registration, and SeCan obtained the rights to them and went through with the seed multiplication, varietal blending with the seed growers. So another roughly 4 years to get us to 2021, where they're releasing commercially on a large scale. So when I say it's a long process, that is a relatively long process. So this is some genealogy just to look back at lines that went into the makeup of Wheatland and Starbuck. You can see on the left side of the screen, it started with a cross of Superb and Ulson. Superb came from the Winnipeg AAFC program. Ulson was a North Dakota line or variety, and they were crossed by Ron DePau. And you can see some lines came out of there, BW 864, 865, Carberry, and much more. So these were doubled haploid sisters. So some of you out there will have a brother or a sister, and you have differences. They're each unique, and each one has some different traits of interest, and some of them have some negatives too. So these were chosen to go forward. Some of them became varieties like Carberry and much more. The other two did not, but they went on. They had value for breeding. So you can see CDC Landmark and CDC Hughes came out of one of them. And Carberry went on, and it's kind of become a backbone. It got used the most, probably because it had the fewest negatives associated with it. So it kind of became a backbone and has contributed to the release of AAC Elida, Wheatland, Starbuck, Russell, Broadacres, and Goodwin from our program. So 1990s crosses to 2021 commercial production. Again, this is quite a long process. It's taken a long time. Targeted diversity was used in parents. There's intermediate steps along the way. There's germplasm exchanges amongst breeders and institutions. There's many breeders, pathologists, scientists involved along the way, and multiple approaches were used. So going back to the beginning of crossing, choosing parents becomes quite important in the breeding program. And we also often hear about genetic diversity and how much genetic diversity we need. And it's true, there are traits that go back in this diversity globally that we can tap into. So I'll use an example here of a global wheat variety that's being grown, not in North America really, but globally. This is what it looks like. So in the Morden FHB nursery, it is absolutely smoked with FHB. And the Morden rust nursery, it has quite reasonable rust resistance. So it has, but it has poor end-use quality as well. It has an excellent plant type. So we may want to cross with this for its rust resistance. It's genes that it has incorporated for that. But we have a lot of deficiencies to overcome in end-use quality and FHB resistance. Another one is an Eastern Canada experimental line we tested in the Swift Current program. Excellent FHB resistance, but it was quite susceptible for most other diseases, including leaf rust, stem rust, stripe rust, I think common bunt and loose smut as well. They were very poor, but yielded very well and had excellent FHB resistance. So again, another source of diversity we can tap into, but a long way to go with it. So I don't want to take too much time or get into too many details here, and I may have too many slides, but just a note on basic research. When I started in 2011, we did a project where we took adapted CWRS material and crossed them to look at stripe rust resistance within some adapted CWRS varieties. And we did these stripe rust assessments in Canada, Mexico, and New Zealand. We did some fancy genomics work with a 90,000 marker chip, and that was applied to every individual out of those populations. And essentially we identified chromosome regions that conferred the resistance that we were looking for. And there's 42 chromosomes in wheat. That's what this is depicting on the bottom of the screen. And where on those chromosomes is that disease resistance located that we can tap into? So one out of two of the populations, we found this loci, we call a locus, sorry, a region of a chromosome that was in common between two of the populations and conferred resistance to a number of rust, leaf rust and stripe rust races globally. So it's an important place that we'd like to focus in on in breeding. So we talk about translational research. And it's really taking the basic research and moving it into breeding. So we worked with NRC to convert some of these markers. You'll see weird names on there like Bob White and RAC and all these different, those are just genetic markers on a chromosome. So we wanted to make these easy to use in the breeding program. So we worked with NRC to develop this new markers, convert them into a marker system that we call CASP, and you get these nice computer generated images of lines that have the marker or don't have the marker. So that becomes easier for us to use in the breeding program. So we approached Sask Wheat about funding this, and they did, and we're getting to be about a year away from finishing the project. But we basically selected the best lines from each population and intercrossed them with Starbuck. So we developed those DNA markers, and we're able to use those at the earliest generations to enrich for desired genes. And now we have a number of segregating lines with improved agronomics yield, especially stripe rust and leaf rust resistance, and end-use quality. So we're able to find untapped diversity within the CWRS germplasm to bring it together in a faster way, a relatively short time frame, rather than going to necessarily just global diversity that could take more like 30 or 40 years to clean up. We call it linkage drag when we get undesirable things that come along with some of that global diversity. So there's a lot that we can tap into using some new genomic tools. So there is a significant probability that a variety would be derived from this material. So research and breeding together, they're both cyclical. Pests evolve, end-use quality improvements are often needed, and agronomics, we're always looking for more yield. So integrating that basic research that we do with what we call translational research, kind of the middle ground, into applied breeding, it becomes very powerful. But teams are required to pull this off, and you really need access to all the tools in the toolbox to do it. So my MSc advisor used to always say to me, good things take time, bad things happen instantly. And I think that's quite true in breeding. A lot of the work that goes on may not be realized for maybe decades, but it's being done with the best intentions in mind. On the bad things, well, COVID hit, and that really threw things for a loop that we weren't really planning on. So we were able to make use of some other research, and I don't want to get into too many weeds here because I'm running out of time, but another topic is genomic selection. So it's basically taking all the data that we're able to generate in the program, the yields and the proteins on the segregating lines coming through the program that often have a parent in common, like carberry, as we saw with wheatland and starbuck, as well as more complex traits like Fusarium head blight resistance and dawn, as well as end use quality. These are the things that tend to be quite expensive to assess in a breeding program. If we can take all that data that we have and use advanced statistical models, and then use a breeder chip that we developed with the University of Saskatchewan. So a pared down version of a chip, essentially. So 5,000 markers per line that we can visualize everything about them. And if you're familiar with 23andMe or Ancestry.com, it's quite a similar thing. You spit in a tube and you send it away, and they'll tell you what your likelihood of having type 2 diabetes is, or if you have certain mutations for cancers, or if your hair is more likely to be curly. That's the same sort of thing that we would do here. We would be able to predict the performance, or more accurately, the lack of performance of lines on average, and be able to discard them more at an earlier stage. And that leads to better varieties. So if we go back to that funnel with those numbers, the F4 generation, if you pull some of those out and you really study them intently and genotype everything in the program, or run those markers on every line in the program at a cost of $30 per line, which is reasonably cheap in a breeding program, we would be able to apply those genetic statistical models back on the overall program and throw out the worst. So implementing genomic selection 2020 was a challenging season. It's not a secret that we weren't able to seed everything we wanted due to COVID protocols and when COVID hit. So we're currently genotyping all the CWRS early generation material. The training set modeling has shown promise for some of those traits like grain yield, grain protein, and grain quality traits. And that image in the background is a lot of the material that we have sitting in New Zealand. We actually left some material in New Zealand in the spring because flights back were difficult and very expensive. So we're reducing our numbers actively right now, so we won't have to ship as much back, and we won't have as much to test in the field this spring. So some good things can come from it, and it's from the foresight and of doing that work proactively on genomic selection. And very importantly, having the support of farmers and the farmer checkoff and the commissions to make a lot of these big projects work. So with that, I'd like to thank the funders, Sask Wheat, the Canadian Wheat Research Coalition, and everyone else who supports the program. It's just a picture we took a few years ago when we didn't have to socially distance in the field with a lot of the equipment that's procured through a lot of the funding we receive. So thank you.
Great. Thank you, Richard. That was a great presentation. And I assure you, even though it says Dallas Carpenter as Richard's name there, that was Richard doing that, not me doing a Richard Cuthbert impersonation. So thank you very much. And we're moving on now to Jim Downey. Jim is with SeCan, and he's going to give you an overview of the newest varieties for 2021. Jim.
Hi, Dallas, and everyone there. Just going to share my screen, hoping that popped up.
Looks good.
Just going to go into slideshow mode here. And thanks, Richard. That was a great presentation. I just want to spend a bit of time and quickly run through some of the newest varieties that will be available in spring of 2021. And just happens that a lot of these are from the Ag Canada Swift Current breeding program that Richard is in charge of there. I'm going to start out by talking about a couple new varieties called one AAC Starbuck and the other one is AAC Wheatland. Both came through the program, as Richard says, at the same time. Both are midge tolerant. So this is just some pictures and just a quick overview of the varieties. That's Carberry on the left. Carberry, as Richard's talked about, came out quite a few years ago, and it was kind of a prototype being short, strong straw with the MR rating for Fusarium head blight. It was not a top yielder, but it was kind of a new stature, and it had the best Fusarium rating available. Very upright variety. Starbuck is just to the right. Starbuck is looks a little bit more like Brandon, about the same height as Carberry, but a slightly floppy head like Brandon has. So Starbuck came through Richard's program. You can see there's some American parentage in the background with Grandin and Caldwell. That's where some of the midge tolerance came from. It's an Ond semi-dwarf, midge tolerant, about 113 to 118% of the yield of AC Carberry. In the Saskatchewan Seed Guide, this is our top yielder. At this point of all the varieties that are available this spring, this will be the top yielder. And maturity similar to Carberry, height just a slight bit taller than Carberry. The FHB resistance rating is rated as moderately resistant, and it has low dawn accumulation. And it's got a good resistance to leaf rust, stripe rust, and loose smut, and has intermediate resistance to stem rust. So overall, a fairly good disease package. It is susceptible to bunt. Lodging is rated as fair and sprouting rated as fair in our seed guide. So that is an improvement on the sprouting rating. Brandon is rated poor. The lodging rating is rated fair, although when I look at the variety, it seems to me more like a Brandon, actually fairly good straw strength in a wheat variety. It does have a high falling number, which translates to that better sprouting rating than Brandon has, and just a little bit lower protein than Carberry. This is just some numbers from the registration trials, just a quick bit of data. You can see here about a 14% better yield than Carberry as it went through the registration trials, and as it's been tested more in Saskatchewan trials, it's held that advantage in that 113 to 118%. You can see it's about the same height as Carberry, and as lodging resistance, at least in the registration trials, very close to Carberry. The lower that number, the better for lodging. You can see also a high falling number over here that translates to pretty good rating for sprouting, much better than Brandon. These are just a few pictures of Starbuck in plots at the Kernan farm in 2017. This is Starbuck. You can see it's shorter than average compared to varieties around it, and fairly upright. Again, Starbuck compared to Carberry under irrigation, not quite at maturity here, but you can see similar height, similar look. Again, Starbuck compared to Brandon. Brandon has a bit more floppy head than the Carberry, and Starbuck looking quite similar here under irrigation and Lethbridge. Again, just a shot of Brandon. This is really the variety that we compare a lot of things to, and we often compare both to Brandon and Carberry at the same time. Another variety here, very similar to Starbuck with a few small differences. This is AAC Wheatland. And there's again another shot of Carberry versus Starbuck. So I should be using the words AAC Wheatland VB and AAC Starbuck VB because both varieties are midge tolerant, and they're both be released as a varietal blend with 10% of the varietal blend being AAC Brandon, and that will be the refuge for the midge tolerance. Just a picture of Wheatland up in North Battleford, strong strawed variety with very good yield back in 2018. This is one of the first years of seed increase. This is a picture of AAC Wheatland VB, and with again in plots at Kernan Farm, you can see a bit shorter than the varieties around it. AAC Wheatland VB from Richard Cuthbert's program, some Carberry in the parentage, and on semi-dwarf, the midge tolerant. The refuge will be AAC Brandon, and Brandon at 10% of the blend. It is in our Saskatchewan Seed Guide, yields about 112 to 114% of AC Carberry. So just slightly lower yield than Starbuck, but again, a very competitive, very high yielding variety. Maturity just 1 day less than Carberry, and again, centimeters taller than Carberry, very similar to Starbuck. One of the big differences with AAC Wheatland VB is that it is a rated intermediate for Fusarium head blight. So not quite as good for Fusarium as Starbuck or Brandon and Carberry. Good leaf rust resistance, stem rust resistance, and intermediate resistance to stripe rust. Another difference with AAC Wheatland VB is it is rated very good for lodging. So it is a stronger strawed hard red spring wheat, stronger than say Starbuck, and it has a good rating for sprouting, again with a high falling number. So there's some numbers from Wheatland coming through the registration trials. You can see even in the registration trials with slightly lower yield than Starbuck, about 12% better than Carberry. Height, it was just a centimeter shorter than Starbuck, very close to Carberry, but a very good lodging rating. The lower the number, the better again, and you can see that it's lower than Carberry. So Carberry is kind of one of our checks. Viewfield's probably our strongest strawed hard red spring, and you can see it's very similar to Viewfield, but with better yield. And so overall, and a good high falling number over here too, and good protein. Just another shot of Wheatland under irrigation. Wheatland looks more like Carberry with a very upright straw type, very upright head. Starbuck a little bit more floppy in the head. I'm going to move on to talk about a few other varieties that are just a year or so older. I want to talk about AAC Elida. This is Elida compared to Brandon. You can see it's a bit taller than Brandon. And it's actually AAC Elida VB because it is a varietal blend with some refuge in there. Should be a Brandon as the refuge. Again, this was parentage was Carberry in the background again. It came through the registration trials about 4% higher yield than Carberry, maturity equal to Carberry, but about 6 centimeters taller than Carberry. This was one of the midge tolerant wheats, one of the first ones with the MR rating for Fusarium head blight, so moderately resistant. So this was an improvement over older midge tolerant wheats that were more like rated intermediate for Fusarium head blight. This one has a good disease package, also with a slightly weaker straw than Carberry, but very good sprouting resistance. I position Elida as a very good midge tolerant wheat, improved yield over older varieties, with better straw strength and very good grade retention, very good sprouting resistance. It actually has quite strong straw, pretty low rating for straw strength right here, but even though it is a fair bit taller than Carberry. So Elida was available last spring for the first year, and you can see here it looks a little bit taller than Brandon, but is very upright. And just going to move on to talk about another new variety called AAC Tisdale. And first of all, I'm going to mention AAC Warman. AAC Warman came out of Ag Canada Brandon. This is just a shot of it growing near Winnipeg in 2017. And this was, again, a VB, a varietal blend. This comes out of Santosh Kumar's program in Ag Canada Brandon. It had some cane in the background and has some Olson also in the background. An on conventional height variety, midge tolerant, refuge to be determined in there. Refuge should be, again, Brandon in Warman. Yield was about 102 to 108% of Carberry, with maturity 2 days earlier than Carberry, but it was 13 centimeters taller than Carberry. So we tend to call Warman kind of a conventional height midge tolerant wheat, but it also had the MR rating for Fusarium head blight with the low dawn accumulation. So it was a very good variety, but is rated poor for lodging and fair for sprouting. So kind of an old school, taller variety, not quite as strong straw as some of the new varieties, but a very good resistance to Fusarium head blight. So this would be a replacement for older varieties like Unity and Jatheria. Here's just some co-op data. You can see the lodging rating is not terrible. It's better than Unity. Unity was an older midge tolerant wheat, so it's better than Unity, but not as good as things like Carberry and Viewfield. For lodging, height, conventional height, a little bit shorter than Unity, but not not as short as Carberry. Just to compare it to another midge tolerant wheat, AAC Getheria VB on the left and Unity on the right. These are two older midge tolerant wheats, and Warman is much the same as them. And this is Unity versus Warman. So sort of like in a family, Warman being having the better straw strength and better Fusarium resistance. Just going to mention Parata. Parata is a fairly new, early maturing hard red spring wheat that comes out of the University of Alberta. That has some Splendor and CDC Go in the background. This is a ond wheat, about 98 to 103% of Carberry, fair bit taller than Carberry, about 9 centimeters taller. Weaker straw than Carberry and Brandon with fair sprouting rating. But the real claim to fame probably is that it is its maturity. It was not quite as early as Redwater, but it's probably one of our earliest maturing hard red spring wheats on the market right now. And good package for leaf rust and stem rust. Intermediate for Fusarium head blight. Just a picture in the nurseries up at University of Alberta in Edmonton. You can see this is Parata in the middle here. This is Dean Spainer, the breeder. And you can see it's quite early maturing compared to all the other wheats around it. So it is one of our earliest maturing hard reds at the moment. Just a picture of Parata being produced, seed increase out in Manitoba. Just wanted to mention Tisdale for a second. Parata and Tisdale is together here in this registration trial data. Parata with 107% of the yield of Splendor. Splendor is an old early maturing variety that was still a check in the co-op back in those days. And then Tisdale with a bit better yield and a bit better straw strength. And but Tisdale's not quite as early as Parata. So Tisdale, again from Richard Cuthbert, this is a midge susceptible, so it's not midge tolerant, and on semi-dwarf. It was very good for Fusarium, MR rating for Fusarium, 2 days earlier than Carberry, but the protein was about a half to 0.6% better than Carberry. So if you look in the seed guide, Tisdale is one of our highest protein wheats with a little bit earlier maturity. Fair for lodging and fair for sprouting, and a bit weaker and a bit taller than Carberry, but really good protein, good Fusarium resistance, and a little bit earlier maturity. So this is Tisdale, again, just a couple pictures, and that's Tisdale versus Carberry. You can see the height difference between the two. And again, another shot there, taller than Brandon also. And just that table for Tisdale, and this is just Tisdale at maturity versus Stettler out in Alberta. I'm just going to mention a couple things about durum. This is AAC Grainland Durum, a fairly new variety out of Ag Canada Swift Current. Has some Transcend in the parentage. Solid stem with sawfly resistance, and with quite attractive yields, about 5 to 10% higher than Strongfield. So this potentially is one of our top yielders in the seed guide. Maturity a little bit later than Strongfield, a little taller than Strongfield, but a stronger straw than Navigator and Strongfield. So good yield, solid stem, sawfly resistance with pretty good straw strength. And has good rating for sprouting resistance, similar to Transcend. We're hoping this variety, it's fairly new, we don't really know yet, but we're hoping it has good grade retention similar to Transcend. So that one is on the market, brand new. This is just some co-op data, 10% better than Strongfield as it came through the co-op trials. Height was just 1 centimeter taller than Strongfield. Improved lodging here versus a Strongfield, and attract decent grain protein. So that's just Grainland on the right, Stronghold here in the middle, and then Defy on this left side. I'm just going to mention a few things about Stronghold too. Just some pictures of the black ons on the Grainland. It can show black ons, and here it's about 36 inches tall on the Regina Plains. Just going to mention one other variety. This one's been on the market a couple of years, but it's very unique because it's it is our strongest strad durum on the market. So this is AEC Stronghold in 2017. You can see the other durum around it is starting to lodge at the Kernan farm back in 2017. But it's very, very upright. And when we go back to the second rep, it's also very upright here with all the other durum starting to flop over. So Stronghold is also solid stem, which gives soft fly tolerance, very good lodging resistance, strongest strad, stronger than Strongfield and Caberry. It's not a top yielder, but when we go under irrigation, it's about 12% better than Strongfield. And it's about 3 centimeters shorter than Strongfield. It also has good sprouting tolerance. So anywhere out of the traditional derm growing area, as we move north from Swift Current, and if we go into irrigation, Strongfield is a very strong contender. I say it's a no-brainer to grow Strongfield if you're growing north of Swift Current or out into the irrigation areas. Rated moderately susceptible to Fusarium head blight. So it's in there with a lot of the other derms with the MS rating for FHB. This is just how it came through the co-op trials. 5% better yield than Strongfield. A little bit shorter than Strongfield with a much better lodging rating and decent protein. So that was just a picture of Stronghold being very upright, nearing maturity, where something like Caberry is starting to flop and go down. So that's about it for my presentation for now. I'll turn it back to Dallas for questions.
Great. Thank you, Jim. So we do have a few questions here. I'm going to go through these in the order that they came in. Rob Graff, our friend from Lethbridge. Rob, you're on, and please go ahead and unmute yourself and ask your question.
Oh, sorry, I don't have a question.
Oh,
actually.
okay. I just saw your hand up. Okay, no problem. I'll move on to the next one. Taylor Kurtenbach has a question. Was there any bacterial blight noticed in Elida this year?
I'll take a shot. We did hear about bacterial blight in some varieties, but mostly out of Manitoba, and I did not hear about anything in Elida, but Richard might have heard.
I can take a little stab. I didn't get to travel to nurseries this year due to COVID, so I didn't get to see a lot. I did talk to a lot of agronomists. There was a pocket in Manitoba, a view field that was noted to have bacterial blight. From the same agronomist I spoke to, they remarked that red berry did not have any, which is interesting because Viewfield and red berry are sisters. They come from a cross of Stettler by Glen. They're doubled haploids. I believe that it's reasonably anecdotal. No studies have been done on this to confirm there is a genetic difference between those. Elida comes from different CWRS genetics. While they're fairly similar, depending on who you would talk to, researchers you would talk to, some would say they're quite incestuous, but from a CWRS perspective, they come from fairly distinct lineages. At this time, Elida goes back to Carberry predominantly, and Viewfield going back to Stettler and Glen. So more research is needed in that area. The assessments for bacterial blight are difficult. There's a lot of variability, and I don't know if we have very good checks or controls for the resistance. And we have no nurseries that I'm aware of in Canada. There is some being done in the northern plains of the US, I understand. So it's an emerging area we need to look into. But I think the question's maybe going back to Viewfield. So Viewfield has been a check in a lot of the registration trials, and the data I've seen come in, while I didn't get to see everything in person, Viewfield has still performed exceptionally well in those trials. So I'm still scratching my head a little bit, and more research is needed, I believe.
Great, thank you. Next question here is from Claire Langlois. Hello, Jim. In the presentation, I think you said and showed that wheatland was a day less than Carberry, but in the table it showed up as equal to Carberry at 101 days. So did I see that right?
That could be. I'm sometimes blend the Saskatchewan Seed Guide numbers and the registration data, and sometimes there's only a half a day difference and we go to 1 day. So they should be relatively close in maturity.
Okay, Thank you. Next question here is from Wayne Truman. How much difference in FHB is there between Starbuck and Wheatland? Will both varieties need a Fusarium application under normal moisture conditions?
I'm going to let Richard answer that one, I think.
If I had to choose for FHB resistance, Starbuck is the winner between the two. It is lower dawn accumulation and lower symptoms for sure, and quite strong. It was starting to border on R, I believe, so it's a strong MR. Wheatland is very similar to Carberry. It's a back cross of Carberry, so 3/4 of its genetics is Carberry, but it is rated as an I in the seed guides. So Wheatland has the strongest straw by far. It's very much like Carberry. So if you were to really push things and put the groceries to it, Wheatland would be the way to go. But if you're looking for FHB resistance, it's straws like Brandon. So I'd have to take that into consideration and pushing it. But with manipulator, while I don't have any data on that and I haven't seen any, that could be a way to go. So
Okay.
hopefully that helps.
Okay, thank you. The next question is from Tyler West. Any reason for the black awns in Greenland, Jim?
The black awns, that's just a trait that comes along from some of the parents. I'll maybe let Richard speak to that one a bit too.
Sure, we really need the Durham breeder here, Yufeng Ruan or Curtis Bosniak, but the black awns are a genetic factor. They can be stronger in some backgrounds than others, but it's genetics, and it can vary in the genetics. So there are some lines that are quite black on. I think Curtis has a line coming up for registration this year that is quite black on and holds it very well. Some of them kind of come and go, but it is a genetic factor.
Okay. Next question here. Elmer Enns asks, we farm north of Saskatoon in the Sask Valley. What variety, Starbuck or Wheatland, would respond better to management that pushes yields? We typically are using manipulator and 2 passes of a fungicide to facilitate yield.
I'll take a quick stab. I position Starbuck probably as you go east of Saskatoon, where you get a higher risk of Fusarium head blight. I think you want to try to grow the variety with the best level of resistance. But under a terrible infestation of Fusarium head blight, you're probably going to have to spray all your varieties. But Starbuck gives you your best chance of getting a decent grade out of that wheat. As you go to the west of Saskatoon or north, mostly as you go north and west, where you get higher moisture and higher inputs or irrigation, Wheatland should stand up the best. So that's kind of my recommendation. So if you're going to be north of Saskatoon and putting a lot of inputs, I would think about Wheatland. And if you're already using a fungicide, then you're not quite as worried about Fusarium. But I would I would just consider, what is your Fusarium risk in the area? If it's high, then you want to go Starbuck. If it's not so much, then you go Wheatland, I guess.
Okay, great, thank you.
Totally agree. Yeah.
Okay. Now, Russell Firsch has put up his hand. So Russell, please unmute yourself and ask your question. Russell. Okay, Russell, we'll get back to you in a little bit here. Brendan Sigurdsson has a question. How many generations can a variety of farm-saved seed be viable?
I'll answer that. I'll let Richard correct me. Richard went through the generations, and once the variety hits about the F8 generation, we call it true breeding. In other words, because it's self-pollinated, that variety stays the same genetically for thousands of generations. So in theory, you could grow it forever. What happens though is that you sometimes get contamination, or you get sprouting in the field, or you want to change varieties. So that's why farmers tend to want to try a new variety and buy seed on average every 2 to 5 years. Richard, any corrections there?
No, let's hit it right on, I think. Yep.
Okay, great. Thank you very much. We'll go back to Russell here. Russell first, if you want to unmute yourself and ask your question. Nope. Okay. We'll have to get to Russell's question via email. Thank you very much. So that's it for us today. Thank you very much for attending, everybody here. And I would just like to remind you that the emails for Richard and Jim will be available. Just go to saskwheat.ca and I'll have them there on the same page as the CCA credits. So, and then speaking of CCA credits, if you're wanting CEU credits, I should say, sorry, for CCAs out there, please just go to our website and go down to the news section, and you'll see the link there, and you can scan the QR code, or you can provide your name and CCA number to Haley, and her email address will be on there. And one more thing before you go, please register for our next Think Wheat meeting. It's going to be phosphorus and micronutrient fertility for wheat, presented by Jeff Shano from the U of S, and that's going to be on Monday, March 22nd at 11 a.m. So again, go to saskwheat.ca and we'll have the information up there for you to register. So with that, I want to thank everybody again. This was a great meeting, and we look forward to having you join us again on March 22nd. Thanks.